Here we begin the reading of the first unit test on the nature of life. You all have worked very hard, and I know that you will do well and do your best. Question 1. The negatively charged subatomic particles are A. Protons, B. Neutrons, C. Electrons, D. Ions. What are the negatively charged subatomic particles? Protons, neutrons, electrons, or ions? Number 2. An ionic bond is formed as a result of A. Transferring electrons, B. Sharing electrons, C. Transferring protons, D. Sharing protons. An ionic bond is formed as a result of transferring electrons, sharing electrons, transferring protons, sharing protons. Number 3. Starches and sugars are nucleic acids, lipids, proteins, carbohydrates. Starches and sugars are nucleic acids, lipids, proteins, or carbohydrates. Number 4. Which of the following are a function of nucleic acids in living things? Store and transmit genetic information, DNA or RNA. They store energy. They form enzymes. They insulate. Which of the following is a function of nucleic acids in living things? Store and transmit genetic information, store energy, form enzymes, insulate. Carbon forms covalent bonds. This means that A. Electrons are transferred, electrons are shared, neutrons are transferred, neutrons are shared. Carbon forms covalent bonds. Are electrons transferred, electrons shared, neutrons transferred, or neutrons shared? All right, you're doing good. I'm doing good. All right, 6. Water's properties of polarity, adhesion, and cohesion are due to A. Covalent bonds, B. Ionic bonds, C. Hydrogen bonds, D. Isotopes. Water's properties of polarity, adhesion, cohesion are due to covalent bonds, ionic bonds, hydrogen bonds, or isotopes. When sugar is dissolved in water, the sugar is the solute, solvent, buffer, suspension. When sugar is dissolved in water, the sugar is the solute, solvent, buffer, suspension. Number 8. Which of these elements is not one carbon bonds with to form macromolecules? A. Helium, B. Nitrogen, C. Oxygen, D. Phosphorus. Which of these elements is not one that carbon bonds with to form macromolecules? Helium, nitrogen, oxygen, phosphorus. Number 9. Which is not considered a macromolecule? Lipid, nucleic acid, protein, water molecule, which is not considered a macromolecule, a lipid, nucleic acid, a protein, or a water molecule. All right, here we go. Stretch. Number 10. Sucrose is a blank made from glucose and fructose. Sucrose is a monosaccharide made from glucose and fructose. Sucrose is a disaccharide made from glucose and fructose. Sucrose is a polysaccharide made from glucose and fructose. Sucrose is a monomer made from glucose and fructose. I'm not going to reread that. You can rewind it if you need to. Okay. What is the monomer of a lipid? Monosaccharide, amino acid, nucleotide, triglyceride. What is the monomer of a lipid? Monosaccharide, amino acid, nucleotide, triglyceride. Alright, time to put on your thinking cap. You are a researcher and are working with people who are lactose intolerant. You are testing a new enzyme to determine if it breaks down lactose faster than lactase. In this situation, what is the independent variable? I'll read it again. You are a researcher and are working with people who are lactose intolerant. You are testing a new enzyme to determine if it breaks down lactose faster than lactase. Okay, you need to identify the independent variable, and you need to identify the dependent variable. All right, number 14. From 14 on, you can answer with a drawing, a labeled drawing, or a sentences, whatever you want. All right, what is the difference between adhesion and cohesion? Number 15. Describe a saltwater solution. Use the correct elements and the type of bonding that occurs, and state which is the solvent and which is the solute. Sorry. Okay. So from 15, you're describing a saltwater solution. Remember, you can draw a picture. Use the correct elements and type of bonding that occurs between those elements. State which is the solvent and which is the solute. All right. 16. 3 more topics. 16. Explain why an enzyme may become denatured. Use the word substrate and active site, as well as what may cause denaturation. You may draw a picture. If so, you must label the key parts. 17. In our lab, Murder and O'Neill, you tested for macromolecules. Remember when we had all those foods? What were the 2 tests for carbohydrates? Okay, you can just describe the test. You don't have to use the names of what we tested with. But just tell us what happened in the test. What we say describe. Describe the 2 tests for carbohydrates. Why did we have 2 tests for carbohydrates? Okay. And then in the lab Need for Speed, explain how you simulated the effect of an enzyme becoming denatured. Okay, to do that, you need to remember, what did we state was the enzyme in that lab? What was the substrate? And what did we use as the inhibitor? Okay, So remembering those three things, how did you simulate the effect of an enzyme being denatured? Okay? So in our lab, Need for Speed, you explain how you simulated. I'll just take that out. Okay. You simulated the effect of an enzyme becoming denatured. All right, remember what was the enzyme, what was the substrate, what was the inhibitor, and how did you simulate the effect? Okay, put it all together for 21. All right, that's it. You did it. Good job. Peace out.